Okay. I think we will go ahead and get started. Can everyone hear me okay? I'm a little bit short, so if you can't see me, I apologize. So for those who don't know me, I'm Kate Still. I'm the scientific director of the foundation. It's been my honor to work with you all for the past 3 years. And I want to give just a really brief overview. This is going to be quicker than 10 minutes and give some time for the next speakers to come up and talk about what we're going to expect in the science and medicine sessions at this conference and how that aligns with our strategic priorities as a foundation, which is really based on you all. So first, I just want to highlight that we do have a strategic plan in research. I see Kathy smiling because this is her son, Cyrus. And our strategic plan is not up here. It has a lot of goals, a lot of objectives, but just wanted you to know that we have been building that. We have a 3-year plan right now. And it's focused on 2 things. Of course, the first priority is to uncover treatments and cures for a brighter tomorrow. So we are working steadfastly at that every day. But in the meantime, our goal is to help you all get through day to day with some of the symptoms and challenges in daily life. So we're focused on today and tomorrow at the same time. And some of what that looks like in science and medicine is for today, that is, sharing medical management guidelines, identifying new areas of need from you all, so elevating your voice, having quick resources that you can use, in the hospital to have recognition quickly from clinicians, and advising on things like follow-up for genetic testing. We have a program for that. And then making tomorrow brighter is a lot of assisting drug development and clinical trials. We leverage data from you all that you have very generously shared with us or other researchers, and working across disorders and also funding research directly. I just want to highlight that we have 5 or 6 new research web pages. These have been a labor of love between myself and Amanda Bergen, who works with me as Science and Research Administrator, and we've been working to put these together to show you really transparently what our strategic plan is. So if you ever want to see all of our research outline, just head to pmsf.org/driving research, and we have a lot of new information there. And you'll see on the side menu that we also have a lot of links for learning more about current clinical trials, or even for our research partners, how they can engage with us better. One thing I also really wanted to highlight is that we have an investment in all types of research and all phases, as well as all types of drug development. We would like to have as many irons in the fire as possible to be able to help long-term with treatments. So we envision drug development as sort of a continuous cycle, whether you're discovering new treatments in the lab all the way to testing either repurposed drugs or new drugs in a clinical trial. We envision that we need to support all phases of that, because even though things in clinical trials right now could be extremely helpful, something that is discovered now in the lab that is not in clinical trials for 10 years could also be really meaningful. So we are invested in all stages and all therapeutic strategies, because people with Phelan-McDermid syndrome their symptoms are variable. So even between people, there might need to be different treatments. And then with the same person, there might need to be multiple treatments. So we really are agnostic as to the stage of research or type of therapy, because we want to move forward this field as much as possible. The last thing I want to highlight is that this conference agenda represents all of these principles and a journey from this earliest stage of research to current clinical care and knowledge. So today is mostly our cure day, And you'll notice that the first session is really focused in identifying new targets in the lab and using models to mimic Phelan-McDermid syndrome in the lab. So stay with us. That talk will be technical, but also very exciting. And then after that, we're focused on things that are still experiments with humans. So clinical trials, new therapies, but also things like the natural history study or the data hub where you all can participate and we can learn from you in real time. And then tomorrow is a care-themed day where we're really going to be leveraging a lot of current resources, current knowledge, things that you can use right now. And a lot of those speakers are going to be sharing with you expertise in specific symptom areas or in genetic counseling, clinical care guidelines that you can take home with you. And Saturday, we're doing a little bit more of a deep dive. So we're going to have a really exciting talk about kind of this whole spectrum of research in Italy, and then also cross-disorder research efforts. So we are really thrilled to be able to provide this. We chose this agenda based off of your feedback, and we hope that you really enjoy it. And with that, I think we're actually just going to roll into the next session. So I'd like to invite up Dr. Chris Winrow, who is a Phelan-McDermid father, but also a scientific advisor and a scientist. So thank you.
Great. So thank you. This session is finding and testing new cutting-edge therapies. So if you're expecting to be here, this is great. If you're not expecting to be here, you should stay, because this is going to be a really great session. I am a father to Jonathan, who is probably going to cheer in the back at some point, not just for me, but I'm sure for all of the he's actually giving me a standing ovation. So thanks. So thanks, Jonathan. So a really exciting session here today. We're going to be starting off with Dr. Hala Harony-Nicolas from Mount Sinai, the Icahn School of Medicine, and she's a professor of psychiatry and neuroscience. She's an expert in brain circuitry and really building a lot of the models to help understand that research, and specifically the work that as it applies to Phelan-McDermid syndrome. Her innovative work uses transgenic rodent models. I'm sure she'll explain to everybody what that means and how that's really helped our understanding of disease and really enabled us to drive drug discovery forward. She is presenting on behalf of Mount Sinai and Dr. Joseph Buxbaum, who many of you know, I'm sure. He's doing incredible work and certainly is a leader in this field. And then after that, we have Dr. Guoping Feng, who's a Poitras Professor of Neuroscience and the Associate Director of the McGovern Institute for Brain Research at MIT. He certainly is a pioneer in doing this type of work, building all of the exciting animal models that have really helped to drive work forward and actually drive clinical work forward as well. His groundbreaking studies have included these animal models, studying specifically the SHANK3 protein and gene, and have been really instrumental in helping us understand neurodevelopmental disorders. He's also going to be presenting on behalf of Dr. Zhang Guo, who unfortunately is not able to join us today due to illness. She's worked with Dr. Feng and is really a leader in sort of the gene therapy approaches. And again, you'll be hearing about that exciting work and how that has impact for our families in a range of different diseases as well. And then finally, in this session, all of the speakers are going to be diving into the critical role that research plays and some of what's called basic or non-clinical research, how that helps to enable clinical studies, develop new therapeutic approaches, and develop ultimately new cures for us and our families. So really, I wanted now to welcome and ask you to join me in welcoming Dr. Harony-Nicolas to the stage.
All right. Hi, everyone. Thank you, Chris, for the introduction. It's actually really interesting to meet with Chris again in this conference. The first time I met Chris was 11 or 12 years ago in this conference, and it has been a pleasure following after the work of the foundation and seeing this family growing. I think back then it was maybe 10th the size, if I'm not mistaken. Maybe I am, but it really grew a lot, and it's a pleasure to be here among you, families, scientists, and I hope, I'm gonna try and put things in easy terms, but if there's anything you don't understand throughout my talk, please raise your hand. I know there's a session after for questions, but I don't want things, to go vague for 15 minutes. So I'm here on behalf of the Severe Autism Center. I do have my own lab. I feel like there is echo. Do you hear that, or it's just me? Okay, great. I've been working on Phelan-McDermid syndrome and SHANK3 animal models for about 13 years now. I've done a lot of work in that field, in that space, characterizing rat models for the syndrome, and with the goal to understand the mechanisms of, how SHANK3 protein works in the brain. Why is it important? What happens if we lose the SHANK3 gene? But today what I'm going to be talking about is more what are we doing at the Severe Autism Center in terms of drug discovery. So it's not about my lab, although I'm really happy to talk about that as well. And if you have any questions about the animal models, and why they're important, we're going to discuss that maybe later. So when we talk about therapeutic approach and drug discovery, it really takes a village. It's not just the scientists, it's not just the clinicians, it's the families, the patients. It starts with you guys coming to us, we understanding the symptoms, understanding the genetics, moving that to the clinicians kind of translate that to us, and we then start asking the basic questions of, okay, what is happening in the brain of kids with Phelan-McDermid syndrome that leads to the manifestations of the phenotype or the symptoms that we see. And then we start asking another basic questions of how can we target those changes or impairments in the brain in a way that we can maybe help improve those symptoms. So it really takes all these three contributors, families, scientists, clinicians, working together. And we call that the translational research approach. I'm sure some of you have heard about translational studies, translational research. Usually it's used in the context of translating maybe basic science to the clinical side or the clinical side to basic science. I see it as a sandwich. We really take the information from the families and clinicians. Then we as scientists focus on this component. I will go over it quickly. And then we bring the knowledge back to the families and clinicians. And this is how the drug discovery process also works. We need the basic information about the disorder or disease, the symptoms. We as scientists dig into that and hopefully bring it back to the clinic and the patients. So when we talk about discovery or drug discovery for Phelan-McDermid syndrome or other neurodevelopmental disorders, we usually start looking for the causatives. And in the case of Phelan-McDermid syndrome, we have some understanding of what is the causative, and there is a genetic component, obviously. So we start asking, what are those risk genes or factors? What are the molecular targets? So if the SHANK3 gene is missing, what is happening inside the nerve cells that leads to the symptoms we see? So those are targets for us, any change in the cell. And then we use models to more understand what's going on. So one of the things is that we use, and of course that's my specialty, is the genetically engineered animal models. So the idea here is that we try to mimic what is happening in the terms of genetics in the patients. We try to mimic that in the animal model. So for example, if we know exactly what is the mutation, we try to put that mutation in the animal model and then start asking question, okay, after we mutated this gene, what is happening in the nerve cell? So the idea here is not to create a model that is exactly Phelan-McDermid syndrome rat or mouse or autistic mouse or rat or whatever model we're studying. It's basically having a tool that allows us to understand the function of the gene or the protein that is affected in the patients, and then using this tool also to screen eventually for drugs in the future. Because if we have a tool and we have some outcome measures that we can use, we can test drugs. Because nobody wants to go and test drugs in the patient before we have any effectiveness in a model and any proof of concept that it's working. So that's why we use those animal models. We also use human neural models, and this is not to say that we are taking the nerve cells from the patients. It is basically, luckily now, as scientists, we have the tools to take blood samples and transform those cells to neuron cells. And then, basically, we have in a dish neurons or nerve cells from the patient that we generated from their blood cells. And those cells will have the same exact mutation that is in the patient, but also the same exact genetic background. So we're basically studying the nerve cells of the patient without taking the nerve cells themselves in a dish. And this is another model that we can use to ask questions of, okay, what is happening when we lose SHANK3? How do these nerve cells behave? How do they change? How do they, the activity is altered? And also we can use neural cells to screen for drugs. Because they're in dish, we can expand them as much as we want, and we can screen as many molecules as we want. It's even easier than rat models or mouse models, although those are my favorite model, but still screening drugs in cell culture is much, much faster. And then once we have a strong target, we can move to the animal models and we can move to the patients. So that's the idea of translational research, and that's what we do at the Severe Autism Center. In addition to the component of having the families and genetic studies, we have a multi-day clinical assessment on the patients that come to us. We do genetic studies, genetic counseling, and some patients decide to enroll in clinical trials as well. On the other side, what we call it the preclinical side is what we as scientists, basic scientists, do. Use the animal models to ask the question, and there's always crosstalk between these two components, the families and clinicians and the scientists. At the center, we also look at potential therapeutics, and we study that in the animal models or the tissue cultures, the neural models, and also try to translate that to clinical trials. Of course, this is a process that takes a lot of years and a lot of work, but that's basically the path that we take. So I just want to mention one effort that we've done also at the Severe Autism Center. It's called the Autism Sequencing Consortium. It's not unique to the Severe Center, but the Severe Center has been a leading of this effort. It's basically having geneticists, scientists getting together. Instead of each of them having 10 samples, 50 samples, 60 samples of genetic material and trying to find what is the gene responsible for autism or neurodevelopmental disorders, those geneticists and scientists decided, let's all work together, have thousands of samples so we can reach what we call statistical significance, meaningful result to say, okay, we're sure this gene is associated with a disorder. So that's the Autism Sequencing Consortium effort that's still going on, and based on that, we have identified more than 100 genes that are associated with autism, and one of them is also SHANK3, that always appears on the top of that list. So in addition to the earlier findings about SHANK3's contribution to Phelan-McDermid syndrome, the gene is coming up again and again on these, we call them high throughput studies, because these are huge studies, exome sequencing, so they're basically sequencing every base pair in the genome. So when it comes to SHANK3, luckily, there has been a lot of animal models coming, whether from the Severe Center, from Dr. Feng's lab and other labs, studying the impact of SHANK3 on the brain. And what I'm showing you here, I hope it's clear, the lighting. So there are two things kind of sitting on top of each other. So there is this part, which we call the presynapse, and this part that is the postsynapse. What you're seeing basically two neurons talking to each other, two nerve cells connecting to each other, right? And this area where they connect, we call it synapse. So if you ever hear synaptic transmission, synaptic plasticity, whatever, it means this region that connects two neurons together. And what we found in the last decade or a bit more even, that SHANK3 plays a very important role in this connection between the neurons, the synapse. And the role it plays, it's basically a scaffolding protein. So it holds many proteins together so this synapse function well. And we've learned from animal work and neural studies that if we take out the SHANK3 protein, then this synapse will not be functional. There will be a problem with synaptic transmission, which is the connection between two neurons and the transmission of signal or information from one neuron to another. So the concept when we were thinking generally about therapies, many times we think about how can we fix the problem. So, okay, there's a problem in synaptic transmission, so let's try to fix that problem. We can fix it in different ways. We can target different molecules here, maybe not necessarily SHANK3 itself, but let's fix the problem. The new era of therapeutic advancement is based on gene therapy, and you're going to hear about this more from Dr. Feng, whose lab is expert on that. And when we talk about gene therapy, we're not talking about fixing the endpoint problem, but rather fixing the initial problem, and in this case, it's the gene. So what if we replace the affected gene with a new healthy one? What if we go inside and fix it? Not even replace it, fix it. There is a machinery, very elegant machinery called CRISPR, and I'm sure Dr. Feng is going to talk about that, that you can target the gene within the neurons and fix it while it's still there. The other approach is, what if we, I think in the previous presentation you saw that we have 2 copies of each chromosome, so we have 2 copies of each gene. And what happens in Phelan-McDermid syndrome is the patients are losing one healthy copy, or it's defective, and there's one other healthy copy that is working well. So what if we increase the amount of that healthy copy? Right? Instead of fixing this one that is impaired, let's increase this one to compensate for the missing one. So this is what I'm going to show you today, and this is a path that we're taking, approach that we're taking in the Severe Autism Center. The other part, I think Dr. Feng is going to cover. So the idea here is that we have the DNA, it's the genetic material, but this DNA doesn't do much if we don't read it, and we call this transcription. So when we read it, we have a molecule called RNA, and this RNA is then again read, or we call it translated to protein, and this is where SHANK3 is generated, right? But this is where the mutation is in the DNA. So if we do gene therapy, gene replacement, we're talking usually about the DNA. In the case of upregulation, what we're approaching here is the RNA. This is the molecule that kind of reads our DNA and tells the cell which proteins to make. And it's not as simple as it looks here. It's not like just straight line. There are like loops and 3D structures. So this is kind of part of the RNA. And there are some very smart scientists that thought, okay, what if we target these loops with what we call small molecules and make them express more of the gene or less of the gene? And that's work has been done by the Disney lab at Scripps. So they developed a tool that they use in ALS. I'm not going to go through this now. We're running out of time, I see. But basically, it's a tool that allows them to target this 3D structure of the RNA and allow them to overexpress, make more of the protein or less of the protein. So they do that based on computational. They have their tools to kind of identify what are those structures, which molecules attach to which structure, and then if we think about the SHANK3 gene, they take all this information and say, okay, based on this information, which small molecule is going to attach to the SHANK3 RNA? And will it increase the expression? So this is what they do in the modeling process, computational using a software called Informa that they created. But then, of course, you have to validate that in real biology. So the way they do that is they attach this RNA structure of the SHANK3 with a protein that gives us a fluorescence. So it's let's call it green color. And if that protein is expressed, you see some expression. Well, here the illustration is not great, but the white is like basic expression. If I add a small molecule that increases the expression of that gene, then we get more green. If the molecule that I add is actually decreasing the expression, I get less of that gene. So that's a tool that allows us to screen. And that's what we've been doing at the Seaver Center. So there are like different steps that you have to do to validate the process. And we started with 2,500 compounds. Again, it's a collaboration with the Disney Lab at Scripps, targeting what we call the 3' UTR. It's like the end of the gene where there's a lot of regulation happening on the gene. And what we found is that there is 140 hits that show increase of expression. These are all the green dots. Then another screening to make sure that it's specific and not just leading to high expression randomly. So we put the same reporter gene, the green gene, but without the RNA structure. And everything that is here in blue is basically unspecific because it's increasing regardless if it has SHANK3. So following all these screenings, we identified 16 hits, and then the next 16 hits means the 16 potential small molecules that can increase the expression of the healthy gene. Remember, we don't want to fix the impaired one, we want to increase the expression of the healthy one. So we are at this point where we have these 16 potential small molecules, and we are testing dose responses because we have to calibrate the doses that we use. And building on this, we will move to, of course, testing that in biological models, whether it's neural cultures or animal models, to see if it's actually working in the full human. Yeah, so I hope that was my 15 minutes. And I will end with this and say thank you so much for being here, and I'm happy to address any questions after Feng's talk. So my understanding is that we will take the questions after, so I'm honored to invite Dr. Feng. I'll see you again after. Thank you.
All right. Thank you. So thank you, Chris, and I particularly want to thank the foundation and Kate and everyone else, and for your participation. We have known the foundation has been doing tremendous work with your support for many, many years now. So I'm very happy to be here to give you an overview what we have been doing with regarding to Phelan-McDermid syndrome and SHANK3. So my whole independent career, I spent last 20 years, almost all my independent career studying SHANK3 and Phelan-McDermid syndrome using animal models. And thank you. My passion comes from, I actually graduated from medical school in China, wanted to be a pediatrician, and in the hospital as an intern, and realized most of the terrible disorders we don't have a medicine, so I decided, okay, I should go to research. So that's how I end up here. So I always have passion for, disease affect, especially neurodevelopmental disorder affected kids. So what I want, I'm here not only talk about the research in there, but I also represent Shen Gao, was a former postdoc in my lab, developed the gene therapy. You will hear more from Jaguar Gene Therapy, who licensed from MIT and Broad to develop clinically. Really a great team, and they made it possible to be potentially a therapeutics in the future. So I also want to thank all this work made it possible really by a lot of people, especially I want to mention the new Yantan Collective at MIT, which includes 6 centers. One is Center for Autism Research, one is the Center for Therapeutic Development. This was supported by 2 donors, Lisa Yang and Hawk Tan. Hawk Tan, you may have heard of, he is the Broadcom president. Lisa Yang is also a very successful lady, and they donated large amount of money made this work possible. So I want to thank them to make this work possible to even to start. So, Dr. Harony-Nicolas already said a lot of things about, made a great introduction, so I can skip all this, focusing on the meat part of it. And you all know genetics lead to identification of the genes. With genes, now as a neurobiologist, we got excited because once you have a gene, we know how to study them. Whether we can figure it out or not is a different story, but we know how to study them. So the idea is, like you study almost any brain disorders, we cannot get a brain. We cannot get a human brain. And like cancer is a tough problem, but it's a little easier because you always can get a tissue. Because surgeons are very happy to cut them off and give to you. We cannot do that. So we heavily rely on animal models. So that's our, animal models are, great contributor to our neuroscience research. So with animal models, with the why we are so optimistic now compared to 20 years ago, because we have unbelievable number of new technologies developed by all over the world, made it now possible really can look at the brain, and mostly in animal model brain, but it's also now starting to look at it, you can look at it into human brain. And with this kind of study, you can potentially figure out what is the best way or possible way effectively to treat these disorders, which was not possible before. So, Hala already said, the SHANK3 affect the synapse, is critical for synapse development, and many of the genes identified so far in autism spectrum disorder, including SHANK3, affect synapse development or function. So my lab focuses on many other labs, including Joe Buxbaum's lab and many other labs, study synapse development and function with the hope to help us understand how the mutations in these genes affect the brain development and the function. So the SHANK3, you can see, SHANK3 we call it a scaffolding protein, which means is a structural protein. It's there to bring many, many other proteins to synapse. So how big is the synapse? It's about 2 microns in the size. How many proteins there? How many type of proteins there? 1,500. So it's really, really complicated. We always say, if there are 100 ways to build it, there are 100 ways to break it. So that's why the synapse is actually focus of many brain disorders, whether it's depression, schizophrenia, autism, or OCD, or many of the disorders. So this I can skip. You all very, very familiar. What does SHANK3 do? What the Phelan-McDermid syndrome was identified by Dr. Phelan and Dr. McDermid. And our idea was very simple at the beginning. We make an animal model because we know the genetic mutation. We can put any human mutation into an animal model, right? So let's make a mouse model, study it, really, really study in detail. And we might find a pathway that we can develop a drug and treat all the symptoms, correct all the symptoms. So these models, mouse models, are quite good. They have some we call the autism-related phenotypes because they never behave like us. We don't live in a hole and work during night. We go sleep. So, but you can test them. There are some related repetitive behaviors, social interaction differences, sensory differences, and anxiety, all other phenotypes. So then we can use many different ways in an animal model, especially mouse model. We can study them. We can use biochemical method, electrophysiological method, imaging method, all these different ways. And because all these phenotypes we call, in patients we call symptoms, in animal model we call phenotypes, we try to figure out each one, where they come from, right? Because all our behavior are controlled by our neural circuit activity, right? Anything, whether it's thinking, behaving, eating, all the things, you have a correlated neural circuits or a circuit or a group of circuits that functions. Whether you move a hand, millions of neurons are already activated and transducing the signal from your brain to the spinal cord to your muscle. So we thought if we can figure out each one of them, maybe we can find a converging mechanism, say, hey, this is the molecule, why lead to this type of behavior? So we study each one of them, whether it's repetitive behavior, social interaction deficit, or other things. It turned out each one affects different brain region and different type of cells and a different mechanism. So now it becomes really, really difficult to find a druggable target that can correct them all. And you probably, many of your parents, know that there are many, many different symptoms, and a lot of times they're different even from individuals. So that makes it really difficult, because if we are interested in autism spectrum disorder, SHANK3 or Phelan-McDermid syndrome is only one of them. And it's actually an easy one, relatively, because we know the genetic mutation. 70% or more of them, we even cannot pin what is the genetic cause of them or what is the environmental and the genetic combination. So that makes it really, really difficult to find a converging mechanism. You have a drug target, you give a drug, and you will correct of them. So that leads us to think about how can we really develop therapeutics for this type of disorders. And we think there are two, probably after many years, many of us in the field, for most of the monogenics, because of the genome editing gene therapy technology development, the best way, if it's possible, is probably correct the genetic mutation. Now, the mutation you can correct it at the DNA level or at the RNA level, how I just mentioned, right? So there are many different ways, because each gene do many, many different things in different cells at a different developmental time. The gene may do, during embryo is different after birth, because your cell now needs to do a different thing, just like us, right? You're at work or at home, you do different things. That's all kind of similar idea. And so if we correct the genetic mutation, then we have a hope to correct all of them. Because if you have the right gene, we know we are all totally fine. So that's the we're thinking. But how about for the 70% of the rest, autism spectrum disorder? You can, because we couldn't even, they are not caused by single mutation, so it will be very difficult to do gene therapy. So as I mentioned, almost all our behavior controlled by circuit activity. So if we can identify, circuit defect, then we can at least to identify circuit-specific drug target, then can correct some of the most debilitating symptoms. That will dramatically improve the quality of life. So the whole goal is how do we our research to help develop things that can improve the life of the patient and the family. So I'm going to use two very simple examples that both are SHANK3. Right. SHANK3 is a neurodevelopmental disorder. One of the key questions, if we want to do gene therapy, can we correct it, reverse it after the birth or after you already have the symptom? That's usually the diagnosis done. So we did a genetic study in a mouse model. Basically, the mouse grow up without the SHANK3 gene, with the SHANK3 gene mutation. Then you provide the SHANK3 after they're at different age. First day after the birth, 1 week, 3 weeks, or in adult, like 3 months. Can you reverse all these phenotypes or defects you see by electrophysiology or by behavior testing? The answer is yes or no. In adult, you can only reverse certain, you can reverse certain phenotypes, but you cannot reverse all. But if you give them early enough, like postnatal 21, which is hard to correlate with humans, probably in the teenage age, you can still correct most of them. And if it's starting younger, you can correct all of them. So that gives us a feeling there is a postnatal, after birth window, you can do gene therapy, you can probably correct, if that is effective, you can reverse almost all the phenotypes. So that gives us a hope, because this is the genetic manipulation you cannot do in human. So we decided, can we develop a gene therapy approach to testing animal models? And so Shan Gao, she's now at a gene therapy company as director of translational research. So I'm presenting her part. So how do you develop a gene therapy? You all know Phelan-McDermid syndrome, many of them have large deletions. So genome editing, the new CRISPR technology, is still not up to that task yet. And also all the mutations are spread randomly on the whole gene. That makes it precise genome editing much more difficult, because you correct the one mutation, you might be only have 10 patients over the world. But overall, there are hundreds of thousands of patients. So we thought a little simpler way just supply, because we know they're all missing a normal copy. Can we supply the normal gene to it? And then you can use it for any of the patient who has a, we call it loss of function mutation, whether it's a single mutation or deletion. So that's our idea. Currently, the most effective way to get into the brain is the user called adeno-associated virus, AAV, which has been in clinical trial for many, many different, hundreds of different things. But you cannot package into it because it's too big a gene. So we designed a mini gene, we call it mini shank, to include all the important functions of the gene, but cut out the parts which we don't think is critical for the function. This is based on decades of other people's study and including our own study. So then we test them in mouse model, and we found it's actually this gene is functional. The mini gene is functional. Use a mouse model, we can inject through the an IV injection, and you can get into the whole brain. And then you can wait for 2 months and test their function, right? So we found it's indeed either by electrophysiological method measure the synapse function or behavior, it can correct everything, even after postnatal day 14 or post day 28. So that gave us a hope. Our main worry at that time is mice are great models for biomedical research, but their brain is quite different from ours. We are not only bigger, but smarter than, I hope, smarter than the mouse. They do some things better than us. But for gene therapy, if we want to really translate it into humans, we want to make sure it's safe and effective. So we have been thinking that for a long time. For the past 10 years, we decided that to really make it increase the translatability, we probably need a better animal model. What else is closer to human? So we thought about non-human primates. So we're working on called marmoset. It's a very small, from Brazil, non-human primate. Their brain structure is much similar to humans. So we spent almost 10 years to do this with the support from many of the donors. And we made the mutation found in similar to the human Phelan-McDermid syndrome, and allows to test them. You can see these are very small compared to their cage, so they have all naturalistic behaviors, right? So I'm not going to details. With this kind of behavior, you can use machine learning-based automatic tracking. So what we found is that they indeed have behavior similar to, more closer to human patient. And I'm not sure why the slide is not working. So we started to do gene therapy. What we did is to do 3 ages. 3 months old marmoset and 9 months. 3 months old is about a 3 to 4 years old baby. That's much easier to correlate. And the 9 months old marmoset is about 10 to 12 years old kid. So we did both gene therapy. We found it can correct almost everything in these monkey models. So that's how it give us the confidence. So this is how well they behavior like a normal monkey, right? The gray is normal, middle is the SHANK3 mutation, blue is the gene therapy group. You can see they can correct almost all the behavior. And this is their cognitive function. They like touchpad. You put a touchpad in their home cage, they will do things. They will touch the right spot to get a reward. They love marshmallow, so they give a little teeny tiny marshmallow. They keep doing. You can see they can do hundreds of times. And wild type, down the top, normal monkey, the blue is correct, red is a mistake. You can see wild type, they do most of them very, very accurately. But the bottom is the SHANK3 heterozygous. Now, this is a very same genetic mutation as in human patient, and you can see that it's actually doing at the beginning do many more mistakes, and it's continue to have mistakes. So that, but with gene therapy, you can actually correct all these defects you see. Now, all these are behavior. Can we really tell? Did you change with gene therapy brain circuits? As I said, all the behavior are controlled by circuit activity. So you can do functional MRI just like in humans. I don't know whether some of you have been through the fMRI. And you can tell in the SHANK3, right, mutant, you can see on the top left, there is a defect in the brain circuits. But when gene therapy, that's a red spot, that's the defect. But at the bottom, when you use gene therapy, all these are corrected. They restore like white. That's why how this gene therapy gave us hope that it might work in human patients. Of course, the final decision has to be tested in human patients. Now, these monkeys have been with gene therapy more than 2 years. They're totally safe. We have not behavior normal. We have not seen any abnormal side effects so far for more than 2 years now. So we are very hopeful this can be translated with the effort from Jaguar. Maybe we can translate it into benefit human patients. And so in the last few minutes, I just want to say many of the disease, ASDs cannot be treated with gene therapy. But what do? We're not going to give up the majority of the patients, which is about 70% of them are ASD patients. Some are very severe. How can we improve their quality of life? So, as I said, all the behavior are controlled by circuits. If we can figure out the circuits, then we can control the behavior, we can correct them. So I'm going to use a very simple example. In many of the psychiatric and neurodevelopmental disorders, they involve cortex to the striatum, thalamus, cortical, these big circuits. And if we can figure out the defect, we actually, as a neurobiologist, we can find a way to correct them. So again, use Shank3 mouse model to start. We found that in Shank3 mouse model, if you present your stimuli, sensory or social, in this case it's social, and you can see the bottom is the Shank3 mice. Their activity really rank up, and they overwhelm the brain. So that's very common in autism spectrum disorder, right? So, and if we can correct this, then you can predict their behavior will be normal, which is, so we are here identifying the target. So this is the most left is the before the drug, the normal mice. Bottom is Shank3 mutant mice. You can see the brain activity dramatically increase. This is use a new technology called focused ultrasound imaging. And it's very new in the last few years. And then after 2 hours of our drug compound, you can see, and even 7 days afterwards, you see still almost normal. So that means this could be a potential target for us to develop a drug that can control the behavior, correct the behavior without the gene therapy. Of course, there are other things. This is only one of the symptoms, right? But if you can correct some of the most debilitating symptoms, and again, these patients cannot have gene therapy because we don't know what gene causes them, then this is the next approach. Similar to many other small molecule drugs, that in the future we can develop circuit modulators that can really improve the symptoms and improve the quality of life. So I'm going to stop here. I particularly want to acknowledge the Tan Yang Center for Autism Research, supported by Lisa Yan and Hawk Tan, also the Yang Tan Center for Molecular Therapeutics in Neuroscience at MIT, and many other donors and funding agencies to help us to do this work. Thank you.
Hi, everyone. So I'm just running around because we're going to do questions now. Thank you, Dr. Feng. That was excellent. So if you could both go to the stage, I will give one of you an extra mic. And we are going to pull up a polling software for questions. So you can all scan. Hopefully you can scan it. It's a little bit small. The QR code, let us know if you can't. And if you can't, you can also type in pollev.com/PMSF1 and enter your questions. But you can also upvote questions. So if you see a question that you like, please like it, and it'll be more popular, and we can know to ask it. And just shout out if you're having trouble. Maybe we can see if we can enlarge the QR code. Got it? We can't? Okay. Has anybody been able to scan it in yet? Okay, good. And I'm just going to give it a couple minutes to collect some questions. We also have questions online. So we will add those in, and I will choose some of the top ones, and either one of you can answer those. So thank you both so much. Also, a quick timing update. It says on the agenda that the next session starts at 11:15. We are going to do a little bit of a longer coffee break and start at 11:30. This is especially important for Dr. Liza Squires, who will be introducing the next session. We're going to start at 11:30, so we'll have a nice long coffee break in between. Wow, this is working well. We already have questions with 13 likes, so I think I'll just start and we'll see how many questions we can get through and we'll take a break. So this one is for Dr. Feng. In the non-human primate models, is there any knowledge of whether this improved muscle tone or seizures? Are those things that you look at all?
Yeah, that's a very good question. So we have not seen any seizures in either mice or this monkey, non-human primate models. We did test their muscle tone, and by pulling, they like to pull, and they do have slightly reduced the muscle tone, but not as probably as severe as in humans.
Thank you. I see a question about if it would be possible to get copies of the slides. I'm looking for Kelsey or somebody, but I think that we can probably do that. I think we'll have the recording saved, and then we could maybe ask the speakers if they're willing to share.
Yes.
Is it possible to scroll down to see? Or maybe I can stand back there. Perfect. Treatments have been discussed for kids. What about long-term treatments for adults with PMS? And as a reminder from my overview session, there is a lot of drug development going on. There's a lot of testing going on. So this is not everything, but I will field that to either of you to answer.
I guess, again, I'm here to represent the Seaver Center. I think, I don't want to say something that is not accurate, but if I'm wrong, correct me. The treatments that are being tested is not only for kids, and I think the Jaguar experiment with gene therapy is going for the adults, but has been recently also approved for 2-year-old kids. So it is kind of targeting both developmental stages. And when we study Phelan-McDermid syndrome, we always keep in mind all developmental stages. Of course, we consider what we call critical periods. So sometimes we would think, the brain might not be as plastic as we wish, so we might not be able to change things later. But we have evidence suggesting that in some cases that's not the case, and there is potential to change brain function later in life. And that's why those trials are also happening in adults. So I wouldn't say it's just targeted for kids. Please correct me if I'm wrong.
No, correct. So for all the circuits manipulation, use a small molecule to modulate the circuits to treat the symptom. This all will work in all the ages, including adults. So, but for gene therapy, our prediction based on mouse model is younger will work better, but adult will still have an effect. And we are now testing in non-human primate model in adult monkeys to see how effective that is, but we don't have that data yet.
Thank you both. I'm over here now, sorry. So the next question, I can't fully see it, but I think it's a great question, are examples of symptoms that were improved. So this is for both of you, for anything that you're testing, either in animal models or in the lab. What types of things have you seen improved? I think you both have shown some examples, but if you could give a summary.
So I'm going to focusing on the marmoset model. Many labs, Joe Bucksbaum lab, my lab, Yonghui Jiang's lab at Yale, all did the mouse model. And so in in the non-human primate model, we used, in mice, we have to use homozygous mutation, which means all the SHANK3 protein is gone. But in patient, you actually have a normal copy. In the monkey model, the reason we use homozygous in mice, if you only delete one copy, it has very little phenotype. It's almost like a normal, so it's hard to study. But in monkeys, we completely mimic a human condition, one normal copy, one mutant copy. In these monkeys, the most significant phenotype we found is they have dramatically increased anxiety. They are jumpy to anything you present to them, and just very easy to get irritated. And then they have a certain restricted interest. For example, touchpad, correct? They only want to touch one spot. They don't want to go wandering other spot. Y-type monkeys, they will touch everywhere to get reward. No, they only, and you force them to touch this still. So this is another. Then they avoid social interaction. We don't know that is because they are purely social or purely because they don't like anything new present to them. Anything, new toy, new screen, anything you present new, they will go touch it, but most of the time they will stay back. Then they wonder what they will touch it quicker than they will. So they spend much less time. So we did a video, automated video tracking in their home cage. If two marmosets are normal, Y-type, we call it normal, you will see they spend a fraction of time near each other social. They groom each other, they stay there, they look at the same way, and sometimes they're jumping around apart from each other. You have 2 peaks of time. But if you switch that to one is the SHANK3 mutant, you only see 1 peak, which is they apart from each other. They don't stay closely social. They try, so that's because caused by the SHANK3 mutant. So all these phenotypes can be corrected, including their cognitive testing. Now, one thing we did not see a correction is a very complicated, much more complicated cognitive function. So, for example, you change the screen with a flashing density, and the mom has to figure out, to what degree you can, which is denser than other ones, then you get a reward. These more complicated things, we actually cannot correct it. We don't know that's because we did not get into enough neurons, or that's because they're purely developmental. We put into even 3 months, which is 3 years old, 4 years old, is too late. And that we don't know. But there are few things, very few things we cannot. But the most basic things, we can correct all of them. Anxiety-like behavior, sensory oversensitivity, repetitive, cognitive function, all these we can actually correct them.
Thank you both. I have a specific question for each of you. The first one is for Dr. Feng. It asks, I remember hearing that having too much SHANK3 expressed could be an issue just as much as having too little. Was this studied in the primates? And what did you see to determine the dose that is most effective? I also want to give a reminder for everyone that Jaguar Gene Therapy will be presenting later today in our emerging clinical trials session. So I am skipping over a couple of the questions that have to do with timeline of clinical trials and things like that. But for the non-human primates, would you be able to answer that, Dr. Feng?
That's a very good question. Yes, if you have, especially in mouse models, have been shown by Huda's August lab at the Baylor College, if you overexpress the SHANK3 from the germline, very beginning the development, and in all the cells, you will have a hyperactivity-like behavior in mouse model, right? But in our case, if you with dose for gene therapy, up to we have done about 30, 40, or 50% of neurons, we did not see any overexpression. And so in the monkeys, I've showed you, we only be able to get into 15 to 20% of neurons. In this case, we did not see any overexpression effect. There are probably 2 main reasons. One is 3 main reasons. One is it's a synaptic protein, so we call ourselves lucky, because it's not like transcription factor. It can keep working. For synapse, you have 1,500 type of proteins in at the synapse, and there is a slot for each one. We call it a slot theory a long time ago. And if you have too much, you cannot get in because everybody has their position. If it's full, no one can squeeze in. So that's one advantage. The other is SHANK3 mostly postnatally, right? So if we give them later, and the circuit's already formed a frame, it's harder to change its function. The third is we're not getting into every single neuron. Now, in the future generation, can we change that or improve that, just like next generation medicine, to control it will not be overexpression in all the cells to the wild type level? It is a possibility, but no one has the technology yet. But in the future, I think it will, maybe 5, 10, or 20 years down the road, someone will develop a technology that can cap it, that will not overexpression at all. But it is a concern to many of the gene therapy. It will be a slightly concern to SHANK3, but so far in the monkey model, that's why we do monkey models, not only mouse model, we have not seen that side effect.
Thank you. And my next question is for Dr. Harony-Nicolas. It asks, you stated in the presentation at some point that you narrowed down to 16 or so small molecules. Are you still looking at those to see which ones can help increase the production of SHANK3? Or probably a more general question is, do you continue to follow up on hits that you get long term?
Yes, definitely. That's the goal. So at this point, in this kind of process that the Disney lab applies, is you start with a large number of small molecules. So in this case, we started with 2,500. And then you start running different filtering processes until you get to the ones that like the most confident. It still doesn't mean that these are gonna all work. That's why we have to test things in the lab and in animal models. So the next step, I would say the step that we are at now is that we have those 16 potential molecules that have the potential to increase the expression of the healthy copy of the gene. And right now what we're looking at is testing which dose is the best to get to the levels that we look for. We don't want to have much more or less. We want to have a level that is kind of what we expect to see in normal cells. Once that is established, the goal is to move to models, whether it's neural cultures or animal models, and to see if in real life, in real biology, these things are working. So right now we tested that using an assay. It's kind of artificial because you're attaching a molecule which gives us the color, to a small piece of the RNA of the SHANK3. But we want to see what happens in real life when there's the full gene, the full SHANK3 gene, and we put this molecule in neural cells. Right now, what we've done is not neural cells, by the way. It's called HEC cells. So we want the next step is actually to see what happens in the levels of what we call endogenous SHANK3. So this is the actual SHANK3 that is expressed in the cell. And if we're actually able to increase the levels using one of these 16 hits, so that would be our next filtering step, taking that molecule forward for animal studies and then eventually to the clinical studies. Yeah.
Thank you. I wanted to address a couple of themes of questions that we've been getting as I was scrolling through. There were a number of questions about improvement of specific symptoms, especially neuropsychiatric illness related, and I think this is all to be determined. Either speakers can jump in if they would like to, but I think we've heard from them what they've been able to see in models, and then as we move to clinical trials and some of these things, we can hopefully see what symptoms are improved in humans. And we got a question about, is there a clinical trial registry to be informed when these things come online? That is all coming through us, coming through the sponsors of the studies, such as Jaguar or Neuron, if you hear from them. And we post these things, we direct email, these are on social media, this is in our newsletter, but it's also on clinicaltrials.gov. So you can find all the information there when a study starts about what you expect to find in the study or what the outcome measures are. So all of that is on our new clinical trials page under research. And I think we'll do one more question and then we'll take a longer coffee break and come back at 11:30. So a couple of the questions have to do with different genetics in Phelan-McDermid syndrome because we do have heterogeneity, and people with larger deletions might have more likelihood to have certain symptoms such as kidney disorders or hypotonia or things like this. And we also have ring chromosome where you could have structural instability as well. So do either of you have any comments when you're looking at genetic approaches that are specific to SHANK3, if you think it might still help folks with other genetic underpinnings? And then is it possible that, is it possible that in the future, other genes that are identified as huge and important could be targeted also? That's for either of you, sorry.
Yeah, this is a very good question, a tough question. So it is, SHANK3 gene therapy, gene replacement, for the SHANK3 gene itself, whether small deletion or large deletion does not matter. That's why it's a single point mutation. That's why we designed the gene replacement approach. And there are potentially conditions that a point mutation is not a deletion, that only lose part of function. Then you have to think about it. In that case, you have overexpression. For other symptoms, it's Phelan-McDermid syndrome. In that area, there are 3 or 4 other genes. The SHANK3 gene therapy, if the symptoms are caused by other genes, it will not be corrected by SHANK3 itself. SHANK3 is most expressed in the brain. They have way lower expression in other tissues. Unless that's caused by SHANK3, it will not correct them. In the future, it is possible you correct multiple genes at the same time. Will you be able to put a large piece of deletion back? It's a really, really difficult problem. I don't see it happening in a short time, in the near future. But to correct more than 2 genes at the same time, it is a possibility in the future with different approaches. So,
thank you so much.
Including.
Thank you for, I don't know if you wanted to add anything, but thank you for a fantastic session, and thank you for covering these questions.